Has anybody ever seen this? So just watch it. I'm just going to turn the voice off because the voice isn't on it. That's learning. Is that the craziest thing you've ever seen? So when those neurons come together, what's happening is this neuron is secreting some chemicals, and this neuron is sensing those chemicals. And so when those chemicals will come together to form a synapse, when those neurons come together to form a synapse, now they have a connection. And that connection is learning. And so every second that you're learning is a neural connection, is forming that neural pathway between two neurons. And so if you remember the word Spitzenkorper once, you know, those neurons will make the connection and then they'll do something else. But if you strengthen that connection, then that connection will be made long term, or can be made long term. So really it's so much about repetition and keeping that neural pathway strong with all of your information, and it will like absolutely help just to repeat. Yeah.
So when you hear the word Spitzenkorper in that example, it's like when your brain thinks that it heard the sound that resembles that, does it like send a signal to that specific neuron, and then the one that it's connected to has the meaning? Or I assume it's significantly more complicated than that.
Yes, yeah, it's so complicated because what your brain is doing is it's including auditory, it's including visual, it's including like spatial awareness all at the same time, and so it's more than just one neural connection. But that neural connection will come together with an auditory learning and also with an image in your mind, so you have this like visual representation by triggering those neurons so that you have some sort of picture of what that word represents. So it's like incorporating several parts of the brain. And then the more that you understand how simple it is, it's so crazy how complex it is. It's so crazy because the complexity is just out of control, but at the same time, you know, really it's a sodium message, then a calcium message, and a neurotransmitter message, and that neurotransmitter is telling. It's so wild. Yeah.
That doesn't look like a fact.
Yes,
like
right. And like, how does your brain form an image because of a neurotransmitter being released into the environment? Like, blows my mind. I do not get it. Yeah.
Do they think that one neural pathway is associated like one-to-one with a thought? Or do you think that that neural pathway can be used for lots of different ideas?
Oh yes. So yeah, as far as I know, it's not one-to-one, that there are different things that your mind will associate things with. So like your mind will associate, like maybe even without bringing it in the forefront, your mind is thinking like plants, or your mind is thinking, what does Spitzenkorper sound like? How does that word associate with other things that I've heard or listened to in my life? Like, so there are like associations, but yeah, I don't know. It's crazy. It totally blows my mind. But the reason I love to show this is because learning is a physicality. There's something going on in the physical brain, and so. You know, you can say like, oh, I'm not very good at that, or oh, I'm not understanding it, or oh, I don't get it, what's wrong with me? But really, it's a pathway, and just strengthening that pathway, taking time to repeat the pathway over and over so that things come into memory. One of my other students in my other class brought up a good point of like people who have like a photographic memory, for example, or, you know, so me, I forget 98% of what I learn in a day, but he said like people who have a photographic memory, I wonder if like their neural connections are stronger with every connection that they make, so they don't have to repeat 5 times for the neural pathway to be a strong pathway. But for me, I have to repeat it so many times for it to be in my neural pathway. But one thing, I'm always on a rant, but one thing about the medical profession too, is if you're going into the medical profession, you're gonna think you're gonna forget all this. You know, you're gonna go into anatomy and physiology, you're gonna be like, oh my gosh, I forget everything from micro, I did so well at it. But something will come up and there are pathways there. Like the pathways still stay if you have done the repetition work, then those pathways will stay. And you know, for me, you know, I learned a lot of this stuff when I was 20 and I was like, oh my gosh, I was so overwhelmed when I started teaching. I was like, oh my gosh, I like pull out my micro books that are like dust covers, you know, I'm like, I forgot so much stuff, but it was in there. Like it comes back and I'm like, oh yeah, I forgot. Oh, I totally know that. And like, so it's in there. And when you get into a clinical situation, all of this will help, even though you think like, oh, it's not useful. It really, even you understand more fundamentally what bacteria are, fundamentally what viruses are, even if you don't remember all of the little details. Okay, awesome. So let's move on. Okay, we are going to talk, so we're gonna finish fungi today. I'm gonna see if we have time to get to protozoa, and if we don't, that's okay. So here we are. So we finished metabolism, and we are on DDX and kappa. Okay, so here's the budding scar from yeast infection. Here's a bud. That was the question from the quiz, that yeast have these structures that bud off of their main cell structure that is their asexual reproduction. Okay, so we talked a little bit about transmission. So transmission is similar in a lot of ways between all of the diseases that we talk about. So we're gonna talk a ton about transmission when we talk about protozoa, because transmission is the thing with protozoa. But one thing to remember with fungal diseases and fungal organisms is that transmission, so how do I want to say that? I want to say that transmission is more prevalent in fungal infections because there are fungi everywhere. So remember we said they're commensals, so we have yeast all over us all the time. They say, I thought this was a cool thing that I told my sisters, but they thought it was disgusting. But when a baker makes sourdough bread, they say it depends on the type of yeast that the baker has on the baker's hands, because every person has so many fungal organisms and it makes their bread unique, which they thought was disgusting. I thought it was kind of cool. I don't know. It's like their own little fingerprint, you know, of how that sourdough bread is made. But it's grossing out Dr. Bear.
No, I have my neck is a little stiff.
Oh, yeah.
That stuff doesn't disgust me.
Oh, yeah. Is it grossing you out?
Yes.
Yeah, no, oh, okay. Yeah, I get half disgusted faces, half like, oh, cool.
Some bakers are better than others because they're fungor.
Yes.
It's their microbiome.
Because of the yeast that grows on their hands, yeah. So, fungi are everywhere, and spores are everywhere, and the reason that we don't worry about them very much is because we're immunocompetent, and so our immune systems are strong. We recognize fungal organisms easily. They don't typically cause an infection in a immunocompetent person, but as soon as a patient is immunocompromised, it's a big problem. And then also sometimes it's chronic exposure to certain fungal organisms. So we'll talk more about that. But the reason why crops are a problem or animal feed is a problem is because of the quantity. Because if there is a fungal infection in some sort of plant, well, there's thousands of plants that are mushed together to make animal feed or whatever it is. So it's the quantity also can be a problem, even though a person might be immunocompetent. Yeah.
Just have to check on my knowledge also of how spores work. I thought it was the case that you could wash them off, like wash your hands hands off, but then just because there's so many in the air, they would come back. If you wash your hands really thoroughly before baking bread, would that?
Yeah, so you can wash your hands really thoroughly. I mean, can you get them all off? My guess is no, but, yeah, I mean, you can wash them really thoroughly and that does help. But if you think about it, like, probably every 30 or so agar plates we have in the lab are growing some sort of fungus on them. And so, they try to be as aseptic as they can as lab techs, but just from that and pouring the agar and shedding it'll get a fungal organism on it. And as aseptic as they could be, they're just everywhere. So, yeah.
Would there be like fungal spores and stuff like inside of a fume hood that's like cut off from oxygen? Like the ones like where you they have the gloves that you put your hands in?
No. So those are like they try to make those truly aseptic for like I'll talk about that like boy in the bubble when babies are born without an immune system. Their lifespan is generally about 2 years and they have to live in a bubble environment and still they can't survive long term. But they do filter the air so that the air doesn't have fungal spores in it, so they can hypothetically, but eventually they do get some sort of infection, and my guess is it's probably a fungal infection is what they end up with, unless a healthcare practitioner or a family member or something like that I don't know, breaks the barrier or somehow. But also, they have issues with their own commensal organisms on their own body that as they were born and all that kind of stuff as well. But yeah, all good questions. Awesome. So direct contact or indirect contact, and remember we learned those terms as well. That's up to your memory. Okay. So something to think about transmission, direct, indirect, it's a good review of biological versus mechanical vectors. We'll talk a ton about this when we get to protozoa as well. So fungal growth, fungal mycotoxins, we kind of talked about that. So diagnostic factors are fungal metabolism. So it's definitely something that we think about a lot when we think about where in the world we live, because really it does depend a lot on temperature where fungal organisms will exist and grow. They're highly susceptible to different temperatures. So, that's why they say the best sourdough bread is actually in San Francisco, because it's so humid there and the moisture content and all that kind of stuff makes yeast happy to live in that area. Where here it would probably be more dry. We would just have different fungal organisms that exist here than exist in different regions. So when we think about that for two reasons, we think about the environmental impacts of exposure of our patients, but also we think about internally, like how, where is that fungal organism going to exist and grow depending on the area of the body that the organism will cause an infection. So I'll show you some pictures and we'll talk more about that. So then another diagnostic factor is your patient's history and physical. So H&P means history and physical. And so a couple things you think about is, are they immunocompromised? Or have they had here's immunocompromised, we'll talk about that, but long-term exposure. So long-term exposure is a huge problem for your patients. That's usually where you'll get the information if they have some sort of fungal infection or not. Have they been, do they work in a wet, moist environment? Usually animals. Animals is always a key thing. Oh, you work with animals? Immediately the healthcare practitioner will be like, oh, that's where it's coming from. Whatever it is, whether it's a gastrointestinal infection or something like that, they'll just assume that's where it comes from until proven otherwise. Yeah.
What's malaise?
Oh, malaise is tiredness, like sleepiness. Yeah. Mm-hmm. So we'll talk more about Yeah, so their immune system is working overtime. Somebody that has an infection, their immune system is working, so they're just tired. And then a cough and congestion. So a lot of times because they are aerobic, it'll affect the sinuses, it'll affect the lung tissue. And then our last one is immune status. Immune status is huge with fungal infections, except there's a few exceptions, and most of them are infections of the dermis or the epidermis, so infections of the skin. And anyone can get skin infections from fungal organisms like athlete's foot, like jock itch, like ringworm, things like that. And even if that person is immunocompetent or even hasn't been exposed long term. But generally, if you think about those environments, it's like a moist environment or something like that that caused the fungal organism to grow. So we'll talk more about that. So here are some examples of some immunocompromised patients. I give HIV as an example because HIV patients have a really hard time with fungal infections, but also any kind of immunosuppressive therapy, blood cancers, patients with long COVID, trauma patients, burn patients, transplant patients, things like that. Diabetes, so diabetic patients will have sometimes can get fungal infections really easily.
For fungus specifically, are any of these different risk levels than others? Generally speaking.
Well, HIV has always been a huge problem with because the cell type that HIV infects are the T cells, and those T cells they're called T helper cells, which you'll learn all about that when we get to that section. But the T helper cells help every other type of immune cell in the body, and so when that cell is infected, they have a really low immune system. So HIV patients, it's a very bad situation. I would say probably more so, but depending on like if they were born with a genetic condition, that's extremely can cause infection really easily. Probably the severity of the burn, things like that. But yeah, my guess HIV is probably the most, but immunotherapies, like people that take immunosuppressants will get sick more often, they will, but they don't generally get tons of fungal infections.
Somebody who has their diabetes pretty well managed, are they considered immunocompetent?
They
Or
are considered for the
sort
most
of
part
in
immunocompetent.
between?
Yeah, so if their diabetes is managed, so sometimes what happens with diabetes, as you know, I'm sure, is the lack of circulation. So diabetic patients have a circulation problem, and so because of the lack of circulation, they can have a lot of dead or dying tissue.
Even when it's controlled?
Not as much when it's controlled. When it's controlled, they're usually, I wouldn't want to say 100%, but 100%.
Pretty good.
Yes, and always diabetes is kind of a scary comorbidity for I don't want to say all kinds of infections, but mostly if the diabetes is managed, the patients are okay. If the diabetes is mismanaged, when they start getting cellular death in the feet mostly, then they'll get fungal infections in the feet. They can get thrush or fungal infections in the mouth. They can get, and then they can have issues with any kind of, like, if the healthcare worker has staph and brings staph into the room, or bacterial infections are really scary in that case too, because their body can't fight off their bacterial infection. Okay, so here's our silly mnemonic. Mycopad. I don't know if you have a better one. I'm always trying to think of ways to make all of the fungal infections fit into a category. So here's the little mushroom pad that she's using like a trampoline. So mycopad. Myco means fungal organism. So this is just a little side note that the way fungal infections are named are kind of annoying. So they're a lot of times named after the organism itself, but sometimes they can be given a totally random name. So most of the time it's the organism itself, sometimes it's a random name. But you won't have to know these until the final, these won't be on the quiz. But I'm just gonna go through some of these. So, mucormycosis. So mucormycosis, it was named after what are called mucors, but generally it's just a fungal infection caused by molds. So we'll talk more about meningitis, pneumonia, all that kind of stuff. So mycotoxicosis. So hopefully that tells you what that problem is. It's from mycotoxin poisoning. So this patient, if they have mycotoxicosis, is an infection from the mycotoxin poisoning. Candidiasis, that's named after Candida albicans or another type of Candida. So usually that's thrust, yeast infection that can happen in the pits in the groin area, or somebody can have it invasive. Let's talk about what some of these terms mean when we get through this really quick. So we talked about Candida, that's our note card. It's a yeast. Cryptococcicosis. So this one I hate too, because crypto is gonna remind us of something else later on in the semester that's gonna drive you nuts. You just, sometimes maybe the trickier words, put them all together and what they are, so you can differentiate between some of the trickier ones. So this is from a Cryptococcus yeast. Coccidiomycosis is from a coccidioides, which, remember, we hated that term because it sounds like Clostridioides, but it's not. If you guys are struggling right now and you have no idea what I'm talking about, let me know. This can cause pneumonia, flu-like symptoms. So anicomycosis, I really want you to remember that one, and that one I'll definitely ask you about because it's so common. So it's from they're called tinnias. So tinnias are skin infections from fungal organisms. So there's tinea capitis, tinea corporis, we'll learn what those are. But tinea infections are skin/nail infections from fungal organisms. So we're gonna learn one of these are on the note cards, that we're gonna do one of these in the note cards, maybe today. But these are called our dermatophytes. Dermatophytes are skin-causing fungal infections. So I might have lost you already, just stick with me for a second. So your patients will have all kinds of crazy skin stuff. If you're working in a clinical setting, every 2-3 patients that comes in have some weird funky skin thing going on. So to be able to differentiate between skin stuff is like, it's almost like some magical powers to be able to figure out, like, is that a viral infection? Is it a bacterial infection? Is it a fungal infection? What is going on with your patient? Well, hopefully fungal infections are a little bit more obvious of what they look like. And once you kind of see a few of them, then you'll be like, oh, that kind of makes sense. So we'll go through some pictures of what they look like. But tinea infections are fungal infections. Tons of people have them. Last semester, one of my students came up to me and goes, is that a fungal infection? I was like, yep, that's ringworm. It happens. It's extremely common, especially in kids. So what anicomycosis is every single person in this room will probably get it. We will all have it. And it's about when your patients turn about 60, 70, 80. And it's when their nails get really thick and they have that yellow tint to them. And so that's called anicomycosis, or sometimes it's called tinea unguium. And that is an infection under the nail beds. That's a fungal infection. And even though we will all have it when we're older, some people get it when they're younger from getting pedicures or they had some sort of scratch that got infected, and then they ended up with a fungal infection. So it's common, it's extremely common. It's very, very, very common in older patients. Some people get it in their nails, even though it's not very common to get it in their nails because we wash our hands a lot. Our hands are dry a lot. They're in a clean environment. Our feet are moist. They're just harder to protect from getting spores and getting exposed to fungal infections. So that's called onychomycosis, or it's called tinea unguium. It's a nail infection. I have pictures of it, just for fun. Like you want to see that, but it's okay. We can get used to it. So pneumocystis pneumonia. This is called PCP. Pneumocystis pneumonia is getting a lot more common, especially in it is only in immunocompromised patients. It's from this pneumocystis yeast, and it's a pneumonia that's been going around as a nosocomial in hospitals. And then also, I didn't add it on here, but just pneumonia in general, pneumonia, meningitis, as we let me talk about that in a minute. Let's just keep going through this. So pneumocystis pneumonia, aspergillosis, we know what that is. That's from aspergillus. And then dermatophyte infections. This is one of our note cards, so we'll be really comfortable with this. Capitis, corporis, cruris, pedis, unguium, and versicolor we won't really talk about. Versicolor is a different organism, but they're all fungal organisms. It's this microsporum, epidermophyton, and trichophyton that are all dimorphic fungi that cause tinea infections like ringworm, jock itch, and athlete's foot, things like that. So here, let me just talk really quickly about, I kind of like this picture, I guess. I was gonna just kind of draw that on the board. But you have your patient, and you think about like, okay, here's my patient. They're sad because they have an infection. So when you think about infections, infections don't really just come from nowhere. They came from somewhere. Where did it come from? So in fungal infections, because if it's a mold, then it'll be some sort of respiratory infection, or it can be a skin infection because it has to have access to oxygen, right? They're aerobic. So pneumonia is common, skin infections are common, any kind of respiratory, sinus infection, things like that. But what can happen is if that hyphae or the yeast will dig in eventually and get into the bloodstream. So if those hyphae get into the bloodstream, or if the spores get into the bloodstream, then that patient can get an infection, can have like sepsis or a deeper infection, invasive infection from it digging through into the bloodstream. So can molds really survive in the bloodstream? Not really, they're not happy in the bloodstream, but also you don't want hyphae running around in the bloodstream. And if they meet up with any oxygen, they'll try to set up shop even though they don't do a very good job at it. But you don't want anything in the bloodstream, right? So here, if you look at like a sinus infection, that makes sense. So we'll talk about meningitis, but pneumonia we just talked about. So pneumonia is a huge problem for molds because people are breathing in the spores. Dermatophyte infections are common because it's touching the skin. This is lungs, this is lungs, this is skin, this is lungs, this is lungs, this is anywhere, but that's just the toxins itself. This is lungs, this is skin. So basically lungs and skin are a huge problem, but we do have to consider that sometimes it'll dig deeper into the bloodstream, and sometimes it can get into what's called the meninges. So meningitis, we talked about it a tiny bit, right? So in the brain, the brain up here is surrounded by this covering, and so I don't know if you've taken A&P yet, but there's a covering called the meninges. And it's pad. You don't have to memorize this, just know meninges, because we're gonna talk a lot about meningitis. But it's the pia, arachnoid, and dura that surround the brain and surround the spinal cord, and there's cerebrospinal fluid in there. So there's fluid that keeps the brain floating in the brain case and protects our brain and spinal cord. And that fluid can have organisms in it, which happens with meningitis. So meningitis from fungal organisms is really rare because especially molds don't really like to live in there. Yeasts can, but molds don't like to live without access to oxygen, but they can. And especially if your patient is immunocompromised, then it's more likely to set up shop in other places. So mostly respiratory, mostly skin. It can cause blood infections, which we'll talk more about sepsis. It can cause meningitis. Okay, so here, obviously you don't have to go through and memorize all these, but just lung, skin, just kind of look through that if that helps. Okay, so let's talk about antifungal medication. So we've already talked about this, why fungal organisms are so hard to treat. Why are they hard to treat?
Because of the zygospores.
Zygospores are hard. Sorry, what'd you say, Dr. Borman?
There's so many.
Yes, because the quantity of infection is difficult. What was yours?
I said they're everywhere.
Oh, yes, they're everywhere. Yes, because because they're eukaryotic as well. So, because they're eukaryotes, and so it's difficult to find unique targets, right? So it's hard to find fungal targets that are unique for a pharmaceutical to attack because they're very similar to us. So here's our fungal medications that we're gonna remember, just like our antibiotics. So it might be a good idea to pull your antibiotics back up. I made up this mnemonic. If there's a better one, did I make it up or did I find it? I can't remember. I want to take credit if I found it. I sometimes make up really silly mnemonics, so it sounds kind of like mine, but grow fungi every place around the planet. So that's our mnemonic. Just like antibiotics and what is it? Microbes, what is it?
Antibiotics can terminate protein synthesis.
Yes, nice work. Yes, Candy over here. Good job. Yes. Okay, both of you. So here's our grow fungi every place around the planet. So here's grisofulvin, flucocytosine, echinocandins, polyenes, azoles, terbinafine, and polyoxins, or nicomycins. Those are all the same one. Okay, so let's look at their mode of action. What would some of their mode of actions be? Before you look forward in your slides. Cell wall, what are the cell wall? Chitin. Manoproteins. Glucans. We don't have anything that attacks that. I'll talk about manoproteins too. There's another one. Ergosterol. So we have 4 little tiny targets, and actually we don't really have 4 because there's a problem. I'll show you what. Well, we have a problem with all antifungals. But here, so the microtubules of grisofulvin, so microtubules are what happens in cell division is our cells will attach to the centromeres of the DNA and pull it apart with microtubules. So those are just proteins that help pull the chromosomes apart. But they're a little bit different than our microtubules, but not quite. They're just a little bit more different. So we use that as a target for microtubules, but it's not all the way amazing, and it can kill our cells too. But I'll talk more about that. So DNA replication, this is for an enzyme that's specific to fungi with replication. I don't think I have a slide on that, maybe I will. Here we are with glucans, ergosterol, and chitin. So manoproteins are too similar to our proteins to be a target. So let's talk about these. So here, glucan synthesis, chitin synthesis, ergosterol synthesis, DNA replication, microtubule formation. So if pictures help you, I didn't talk about that, but I do want to talk about it. So antifungals, because we are eukaryotes and because fungi are eukaryotes, an antifungal medication doesn't work very well. It takes weeks and weeks and weeks, if not months, for the fungal infection to go away with medication. So this is kind of a weird question, and you might not get it, or you might not understand the way I'm asking it. What is the problem when you give your patient antifungal medications? Before we talk about resistance, what might be the problem? What is, yeah.
They don't finish taking it?
Yes, compliance. They don't finish taking it. So they won't finish taking it, which is a huge problem with antifungals. It's called compliance. Your patient is non-compliant. They won't follow through with taking the medication all the way to the end. So now we have an antifungal resistance problem, because now we have fungi that can survive, and that you've wiped out half of the population. So now this population that's left over can completely take over, and you have a big problem. Another problem with antifungal medication is that all of these structures are very similar to our structures. So even though they are unique, they're similar to us. All of their structures are similar to our structures. So peptidoglycan is very different than ours. But glucans are similar to ours, or ergosterol is similar to ours. And so what might be the problem when you take an antifungal medication? Yes. So there's a lot of side effects. So not only does your patient have to take it for a long time, but they feel like garbage. So that's a huge problem. Yeah.
How come we can target the ergosterols even though we have them in our own cell membranes, but we can't target the nanoproteins because they're too similar to our proteins?
Yeah, they just haven't, if you're in a pharmaceutical development team, like, how successful is it at killing the fungus versus killing the human cells? Like, how selectively toxic is it to make it to where the patients will actually take it, and how effective is it? So it's just like a development problem. Yeah.
So they both kind of kill off both, but it's just less side effects. For the
Less
patient.
side effects.
Okay.
Yeah.
Why don't all antifungals just target the chitin?
Because I think they have a hard time from 2 seconds I've read about it, is that chitin is difficult to break down with pharmaceuticals. So the pharmaceuticals just don't work that well either.
Pretty tough.
Yeah, tough material, yes. It's incredibly tough material. And so, like, for example, a lot of these have side effects, but this one, amphotericin, is one that we use frequently, and especially for IV antifungals for immunocompromised patients. So amphotericin has the nickname amphoterrible, because the patients will vomit, they'll feel horrible, and then they are supposed to take it for months. So compliance is a huge problem with antifungals. So now we have a problem with antifungal resistance. So Aspergillus is a highly resistant organism. Candida, this is from the CDC, Candida is a huge problem right now. Ringworm, so a lot of times we'll have a difficult time treating anything. So I don't know if any of you have ever had athlete's foot, but when you are trying to treat your patient with athlete's foot and trying to get them to use antifungals, for months they just don't. And then it's in their shower, and then they get it again, and then they, so getting rid of fungal organisms is very difficult. Fungal resistance occurs from inappropriate antifungal prescriptions, from underprescribing, from overprescribing, from, it can get in the water supply, it can get into, we have a huge problem with antifungals with livestock and with crops, because they're spraying antifungals on all of our crops, and then antifungal medications get into the water supply, and then it affects our cows and pigs and chickens, and then they will acquire antifungal resistance and all that kind of stuff, just like how we talked about with bacteria. So resistance works in the same way. There's a mutation that leads to some sort of physical abnormality. So let me show you a picture of it. Sorry, I was struggling with my words there. So they'll have some sort of mutation, so it's a good time also to review your mutations, go back over your point mutations again. Enzymes that will break down the drug, like this is our beta-lactamase example, or beta-lactamase example in bacteria. So they'll create some sort of enzyme efflux pumps. They'll change the structure of the target, increase or decrease the target. So you can use the same 4 that you used for bacteria, just to keep it simple. The same 4 that you used of antibacterial resistance, we can use for antifungal resistance. Efflux pumps, immunity bypass, changing the enzyme, the target, or what was the other one?
Target modification, bypass,
Efflux. And enzymes, right?
Yeah, the way they like this.
The enzyme, awesome. Okay, so just remember them in the same way, it works the same way. So antifungal stewardship is a huge thing, so being responsible with diagnostics, education, training, all that kind of stuff. I don't care that you know any of that, just that's called antifungal stewardship, same as antibacterial stewardship. Or antibiotic stewardship. Okay, so fungal wrap up, that's it. So we did bacteria, we did archaea, we did viruses, we've done fungi. So yeah, another organism down, awesome, moving right along. So they're ubiquitous, especially spores are everywhere. There are fungal organisms that are highly beneficial, even to our human health, highly beneficial. We use them commercially, they're everywhere. An immunocompetent patient, you don't really even have to worry about it that much. Like people don't generally get fungal infections very often unless it's a skin infection. They're saprobic, that's a new term that we discovered, that they will decompose rotting material. We have our same fungal ecology, the terms of fungal ecology. They can asexually or sexually reproduce. They have all kinds of the ability to kind of do all weird things. They are opportunistic in immunocompromised patients. We worry about fungal infections in different environments, secrete mycotoxins, and we can develop antifungal resistance, same as antibacterial resistance. Okay, so let's go. So we have, I don't really want to introduce protozoa yet, because I feel like we're just gonna throw you all off. But the problem with protozoa is that we're gonna talk about it on Thursday. And so this quiz is going to be half fungi, half protozoa for the next quiz. So just try and keep everything separate. I think that is hugely beneficial to you in your studying, is make sure that you know what's bacterial and how it's treated and what their unique characteristics are and what they do, what kind of infections they cause, and completely separate in your mind viruses, what viruses are, what they do, what they look like. Some of the students will go like, you know, like on the midterm, and it was, what is this structure made out of? And some students will write like peptidoglycan. Well, it's definitely not peptidoglycan because this is not a bacteria. So just being really careful that you know what's bacterial, what's viral, what's fungal, and as we move on, that will really help you clarify what's what. I try on the midterms and final not to mix them up too much. I try to kind of keep them separate so that in your mind you're not like, wait, where are we again? What organism are we talking about again? I try to keep them separate, but sometimes it can't really be helped. So I'll just try my best so that you guys don't get confused. Okay, so let's do our note card, and then we will take a break, and then you guys can do some studying, and then we'll meet down in the lab. We did penicillium and aspergillus. Oh yeah, we're on dermatophytes today. So you won't be asked to recognize these separately. Just know that what I want to make clear with fungal molds is that all of the meatballs don't always look like meatballs. Sometimes they look like this. So these are fruiting bodies. And if you could guess, remember we had 2 different types of fruiting bodies. So what were these? Sporangiospores, and what were these? Conidiospores. So what out of these 2 would you guess those are? Yeah, those are sporangiospores. So those have spores inside the spores. So yes, I mean, they're not really, don't really look like spaghetti meatballs, but the actual sporangiospore itself is what contains the spores. So we have microsporum, epidermophyton, and trichophyton. These are the dermatophytes.
What do we need to underline? Anything?
Yeah, these are all genus names.
Okay, so
Yes.
dermatophytes we underline?
Not dermatophytes, good point,
Okay.
good point. Dermatophytes is kind of just a category we call them in, but microsporum is the genus name. So it'd be microsporum and then several species. Trichophyton, several species. Epidermophyton, several species. Do you guys understand what we were just saying about that?
So, sporangiospore and dermophytes are kind of like interchangeable names for them?
Good point. So sporangiospores describes the actual structure. So these structures, like for microsporum, this is a hyphae, and this is a sporangiospore. So it will belong into the category of sporangiospores because this is what their sporangiospores look like. So it has the little brackets and it'll have spores in there. So it is a sporangiospore organism because the spores are enclosed. But the dermatophytes are a broad category of anything that causes a skin infection. So all of these cause skin infections. So we just clumped them all into one big group of dermatophytes because these are what cause all the skin infections. Does that make sense? Did you have a question?
I was just gonna, so I think I already asked you before, but why do we underline HIV?
We don't.
Oh, we don't?
No.
Okay.
Yeah.
Alright.
Yeah. Yes, yeah, we don't underline agent.
I thought
Viruses, no. Yes, yes, good point. Okay, so what we were saying about that is that dermatophyte is just like, they've clumped together a bunch of these organisms that cause skin infections. So remember the dermis, or the epidermis, is, what their tissue tropism is, what their tissue of choice is. And, then microsporum, epidermophyton, and trachyphyton are genus names. And they have several species within that genus. Yeah? Okay. So like Staphylococcus aureus and Staphylococcus epidermidis are both Staphylococcus genus, and they have different species. Okay. So here's pictures of what they look like under the microscope, like maybe spaghetti and meatballs, but when you look at this under the microscope, you would not be confused at all what that patient had, right? You'd be like, oh, it's not bacteria, it's not a virus, it's not a protozoa, it's gotta be, it looks like branches. Okay, so these are molds, filamentous fungi. Here's the 3 types. You don't have to write them again if you wrote them on the front. They have, why did I do that again? I messed that up again. They have sporangiospores. So these generally are not opportunistic. There are many different species of all 3. So here's all the tinnias. I'll let you write those down and then we'll go through them. Okay, so this is tinea pedis, is the fancy word for athlete's foot. Tinea cruris is jock itch. So it's usually in the bathing suit area of people who wear wet bathing suits for a long time, or athletes who sweat a lot. And, you know, keeping areas dry and moisture free and give them exposure to the air so they're not just like wet and moist in an area, then it helps to prevent tinea infections. Tinea capitis is basically, it's like ringworm of the scalp. Tinea unguium is onychomycosis, that's the nail infection we talked about. Tinea corporis is ringworm, anywhere on the body. It's a fungal infection anywhere on the body. Sometimes it's in the shape of a ring, sometimes it's not. Let me show you pictures of all these. So we're not going to talk about tinea versicolor because this malassezia is a different type of mold. So here's athlete's foot, obviously extreme athlete's foot. People who have extreme athlete's foot, a lot of times, like homeless people will have extreme athlete's foot, or sometimes diabetic patients will have extreme athlete's foot. Jock itch, so that can be anywhere moisture sweating areas. Here's tinea capitis of the scalp. It's usually not this bad. It's usually, it can also cause baldness. in a certain area, the hair might not grow back. This is tinea unguium, so this is a mild case, this is a more severe case. Older people generally have one or the other, either mild or severe. This is ringworm, so that's, sometimes it will grow in a ring, and just like it will on an agar plate. And that's from one of the dermatophytes. So a lot of times, children will get that from animals, so animals can get ringworm, and so they'll get it from petting stray cats, or cats of any kind, animals of any kind. And cats are extremely common, but also from other kids. Like if kids play in a sandbox or something like that, then they can get ringworm. Okay, that's all I showed, that was it. Okay, cool. So, you guys can, we're going to meet in the room. So, this is what I want to ask, Dr. Rosebeer, do you remember last semester I asked them if they wanted 90 seconds or if they wanted 2 minutes? Oh, I thought everybody wanted 2 minutes, but I don't wanna.
I think 90 seconds.
90 seconds, start 90 seconds, and then in the final, if they feel like they need longer.
Go back. You guys all get a chance to go back and look at the questions.
Awesome, okay, so we have 36 stations. Let's see how many stations we have. I just wanna make sure I give you guys enough time. Okay, so we have 35 stations. So 35 times 90 seconds. So it should take us maybe an hour, a little bit more than an hour. So we're going to, let's meet down there at 2:55, is that okay? And then we'll be kind of ready to go by 3. And then 3 to 4, we'll have till 4:30, so we'll have plenty of time if we need it. I just wanna give you guys enough time, but also don't want you guys to be like having to wait here too long if you guys are burned out. So, it'll give you at least 30 minutes to study, so you guys can use the board. You guys can use those, whiteboards back there if you want to. You can go through your notes, you can